Okay, so now our final lecture on cell signaling is going to involve our kinase receptors. So with respect to our kinase receptors, there are different types, and so we'll go through them. Tyrosine kinase receptors are very important. They are going to be involved, for example, for insulin signaling. And then we're also going to talk about our JAK-STAT signaling. JAK-STAT receptors are used a lot by the immune system. We're going to have ser-thr-kinases receptors that are often going to be used for growth factors. So our kinase receptors, you can see here, we have our tyrosine kinase receptor, which is a very large family of receptors. It has intrinsic kinase activity. So we know that kinases phosphorylate. So our tyrosine kinase receptors phosphorylate tyrosine domains, and they add basically phosphates. So they are autophosphorylation. An example of this is the insulin receptor. Okay, I'm going to go into that in just a moment. Insulin receptors. The insulin receptor is going to autophosphorylate. Now, different from that is our JAK-STAT. Our JAK-STAT receptor is very interesting because you have some sort of cytokine that binds to the receptor. And in this case, you do have a JAK, which is a tyrosine kinase domain that can phosphorylate. And in this case, you have STAT, that is always the binder of that phosphorylation. On the other end, the tyrosine kinase domains, you have to always have an SH3 domain binding to the phosphorylated tyrosine. And in the third kind, we have the ser-thr-kinases kinase receptors, and this is a dimer. It's a heterodimer. Both of these are hetero, and this is a homodimer. And basically, you're going to have one dimer or one of the monomers phosphorylate the second, and then these are going to be associated with another protein called SMAD. And the SMAD and STAT are both eventually going to be the proteins that transduce a signal to the nucleus and become the transcription factors. Whereas in the tyrosine kinase, you're going to have several steps in order to get a transduced signal to the nucleus. So again, this is the insulin receptor, autophosphorylating. It's going to bind to your insulin and then transduce a signal. Now here's a diagram that shows exactly what's happening. There's a lot of moving parts here. So we have again our insulin that's going to bind, is shown here. Our tyrosine kinases are going to phosphorylate. You can see there's many phosphorylation sites. And each of these phosphorylation sites can bind to an SH2 domain. So we're going to have our IRS1. So this is number 1. IRS1 is basically insulin receptor substrate 1. And then this insulin receptor substrate 1 has SH2 domains. So it can interact with your tyrosine kinase receptor. And then next to insulin receptor substrate 1, you're going to have this protein called GRB2. So I'm going to treat a little 2 here. So 2 is our GRB2. And this is what's called an adapter protein. And this is going to be able to bind to the phosphorylated IRS1. So it also has SH2 domains. So basically, your tyrosine kinase receptor is phosphorylated. It will interact with IRS1, the insulin receptor 1 substrate, phosphorylate tyrosine on that receptor. The GRB2 as an adaptive protein will bind to the IRS1. And then GRB2 is going to interact with SOS, SOS, and then SOS is going to bind to RAS. So here we have our G protein, RAS, smashed in the middle of insulin signaling. Now, our SOS receptor is going to have SH3 domains. Should put a 3. And it can interact with GRB2 via prolines. So SH3 domains bind prolines. Now RAS, which is going to be my number 4. Now RAS is going to be a G protein monomer, so this is not a membrane-associated RAS, this is a free-floating RAS. And RAS is going to start a kinase cascade by activating RAFT1. Now, as long as RAS remains in the GTP state, it's going to be able to continue to activate RAFT1. And RAFT1's whole purpose is to do a kinase cascade with MEK, ERK, etc. So RAFT1 is, and this kinase cascade is going to end in ERK, and ERK is going to go into the nucleus and become part of a transcription factor. Now, ERK is going to activate ELK, and what's known about ELK is ELK can activate over 100 genes. And of course, these genes are going to be involved in some sort of response that you want to do with respect to insulin. Now, ERK is a family member of what we call the MAP kinase family. Now, the MAP kinases get their name from promoting growth. So this is a mitogen. Mitogen is a very powerful stimulant, activated protein kinase. And MAP kinases are going to be used in growth factor signaling pathways. An example of this is our platelet-derived growth factor, PDGF, platelet-derived growth factor, and epidermal growth factor. And so remember that these are all downstream of RAS. And so RAS is our proto-oncogene. So if you don't get RAS to turn off, these MAP cascades are going to continuously to be turned on, and then you're just going to get constant growth, which leads to cancer. Now, your insulin receptor has something called a branching point. And so an example of this is shown in muscle cells. So basically what's happening on your insulin receptors, the same thing we just said, is that you're going to have your signal transduction mediated through your IRS1, going eventually through RAS to activate your MEK kinases and then ERK, etc. So that all happens. Also at this IRS1, you can also get recruitment of another set of genes, and this is going to activate metabolic responses. So your insulin receptor has two things it wants to do. One, it wants to do an immediate metabolic response regulating enzymes, and 2, it wants to activate those genes necessary to carry out its functions. And all of the branching point happens right there at the insulin receptor, right there from the binding of that first protein, that IRS1. So there is going to be also crosstalk. So crosstalk means that you can have multiple hormones signaling the same cell. And so it's going to be very complicated, and it's really up to the cell and what the cell needs as far as what is going to play out. So insulin and epinephrine and glucagon are not going to be independent of each other. Insulin is going to oppose epinephrine in those tissues. And in fact, insulin is the dominant hormone, so all other hormones are called insulin-opposing hormones. Insulin is able to attenuate the signaling through those G proteins that epinephrine uses by basically turning off cyclic AMP. So our insulin is going to be able to degrade cyclic AMP and convert it to AMP. So basically we have again our insulin receptor, and we have our G protein receptor shown here. And with respect to insulin, we have our IRS1 that's doing its thing. IRS1 is also going to activate protein kinase B, which is going to phosphorylate the G protein receptor, and this causes internalization of the G protein receptor. So basically, you're taking away the receptor so that the cell can't even bind to and respond to the epinephrine. And then also, your insulin can hijack your G protein receptor. So minimal phosphorylation of the G protein receptor tail will lead to another downstream pathway to get your ERK turned on, which will turn on the genes for insulin, not the genes for the G protein receptor. So the insulin receptor and insulin signaling will downregulate the G protein receptors, and while it's going through that process, it will also hijack the receptors and use it in order to amplify its gene transcription. Now another kinase receptor or another kinase signaling pathway is called our JAK-STAT signaling pathway. And so basically the first thing you're going to have is an activated JAK. So our activated JAKs are going to phosphorylate STATs. And STATs, when they get phosphorylated, STATs are called signal transducers and activator of transcription. Right, so STAT. It basically means this equals transcription factor. So all that stuff that your insulin receptor did, going through all that pathway of activating RAS and then activating MEK and then activating ERK and ELK and blah, 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 your JAK-STAT signaling pathway is so easy. Your JAK-STAT signaling pathway simply phosphorylates STAT. Two STATs come together, and the two STATs they join together at their phosphorylation. The SH2 domains, again, same thing that we saw with our insulin receptor. And the STATs become the transcription factor. Now you also can use, again, that SHC that insulin used, and you can activate a MAP kinase cascade as well, which will affect gene transcription as well. But the simplicity of the JAK-STAT is that your JAK gets phosphorylated at the receptor, phosphorylates STAT, JAK gets activated, phosphorylates STAT, and the dimer of two phosphorylated STATs are the transcription factor. Now the next receptor is our ser-thr-kinases kinase receptors. An example of this is TGF-beta, which is transformation growth factor beta, And this is important in bone morphogenic proteins, which control proliferation, differentiation, and cell death during development. There are two membrane-spanning regions or subunits. We have our type 2, and then we also have our type 1, which would be the next one here. So type 2 binds to TGF-beta first, and that initiates type 1 to bind. Type 1 is going to have your phosphorylated serine residue, and then your type 2 receptor phosphorylates your type 1 receptor at that serine residue. And then you have some SMAD proteins. SMAD proteins are very receptor specific, and the phosphorylated type 1 receptor at that serine residue is going to phosphorylate your R-SMAD, or receptor. That receptor SMAD is going to complex with its co-receptor, which is co-SMAD, and these two together are going to become your transcription factor. So these go to the nucleus, and they turn on genes, etc. Okay, and then just a little bit about our calcium calmodulin. So we've talked about calcium calmodulin already. So calcium increases is going to activate its partner, calmodulin, and these are going to bind to target proteins. Some of them are going to be, for example, are going to be kinases again. Calmodulin kinase family are CAM kinases are activated by calmodulin, calcium calmodulin. And so calmodulin is activated only when calcium increases, and then they're going to bind to their proteins. They're going to phosphorylate just like other kinases phosphorylate. They are going to help regulate ion channels, transcription factors, neurotransmitters, all kinds of things. We'll see calcium calmodulin pop up every now and then, but that's basically what they're doing. They're going to turn on kinases, which is phosphorylation. And then finally, termination of the signal. Again, you have some sort of stimulus. You're going to initiate release of some sort of hormone. You're going to bind to some receptor. And at every step along the way, you can have termination. You can have termination of the actual stimulus. You can have your degraded messengers, so your messengers don't even make it to the receptor. Again, you can downregulate your receptor, so that's very common. So after a G protein is activated, it actually gets downregulated and recycled. It goes back to the surface, and if there's no more epinephrine, then it can't get activated again. That's a very common way to control things. You can activate protein phosphatases, which again, these are going to remove your phosphates. You can turn off those GTPases. You can turn off your RAS, hydrolyze the phosphate, revert it back to GDP, and then you can have your phosphodiesterases going to degrade your secondary messengers, for example. And a phosphodiesterase by insulin is going to degrade cyclic AMP from your G protein receptors.